there have been studies as well showing evidence of physiological decline for decades, and in some species, not all, that it will be observed an increased reproductive output with age. Older animals lay more eggs, for instance. So, of course, we haven't studied these species for hundreds of years, but in these studies for decades, and there has been studies done for decades in turtles, suggest that these species don't age. So, I think there's always been an inspiration for me, a bit like birds were an inspiration to the Wright brothers. Because you look at these animals and say, well, aging is not inevitable, it's not universal. There are species that appear not to age, or at the very least, they live way longer than us. So if they can do it, well, there is a way, if we can figure it out how, there is a way of us avoiding aging. Of course, it's much harder than it appears at first, but in theory, there's no reason why we cannot stop the aging process. Now, the other major finding I'm sure you've heard of is that the process of aging is surprisingly plastic in the sense that it can be manipulated by genes and by interventions. So Tom Johnson and colleagues, they were the first to identify a single gene that could alter significant the lifespan and the aging process in worms, in C. elegans. So that was age 1, and I think the current record for a single gene, which also comes from worms, is about 10-fold. What that means is, single gene, if you knock it out, I think it was a knockout, if you knock it out in worms, the mutant lived 10 times longer than the wild type. So that would be like humans living over 1,000 years. Now, of course, mammals are more complex. You don't observe such marked effect in mice. I think the current record in mice is probably around 50%. I mean, single gene, which is roughly in mice, and increased lifespan by about 50%. But the point is that we can manipulate aging. And it's not just lifespan that is increased, it's health span. I mean, it's delay of aging-related diseases, it's a retardation of physiological decline. So it's not just the animals are living longer old, it's that the process of aging appears to be retarded. So we can manipulate aging. I'm sure you've also heard of, there are some mutations in humans that may also alter the process of aging in humans. This is one example. So these are 2 siblings, and as you can see, the woman on the right, she has a mutation in a gene called PROP1. Doesn't matter what it does. The only thing that matters the same is that homolog of PROP1, when knocked out, the mouse homolog of PROP1, if knocked out in mice, extends lifespan. And as you can see, it does appear, well, she looks younger than her actual age. We don't know if individuals with this mutation live longer. But it does appear to have some effect. There's other families with other genes in the same pathway that have been studied. It does appear to have an effect. And I'm also sure you've heard about caloric restriction. Does anyone in the room practice caloric restriction? Okay, so you're not truly convinced of the benefits of caloric restriction. Neither am I. I've never done it, never tried it. I do know people who've been on caloric restriction and tried it. So you know the caloric restriction. So this is a very nice study in rats, I believe. Probably from a farm in San Antonio, I would think. So you have a group of animals that have lived. That means they can eat whatever they want when they want. You have a group of calorie-restricted animals, and there's a very clear effect on survival. They clearly live longer. Not only they live longer, but their aging process, again, appears to be delayed. They're smaller than the wild-type mice, but they also have fewer diseases, like metabolic syndrome. They also have less lower incidence of cancer, etc. And
sorry, this data is from 1935?
No, this is from Edward Massaro. He's San Antonio. I think this data is from 1978. Oh. It's one of the, if I'm not mistaken. So it's probably dated from the 70s. Okay. So this was a nice study they used.
Yeah.
Well, this does not correspond to this.
Oh, okay.
This is the original demonstration. Well, the original finding of caloric restriction from McKay and colleagues at Cornell. But they used very few animals. In fact, it's fairly ironic because it's been pointed out by numerous authors that if they tried to publish these findings now, it would be rejected. every single journal because they didn't use enough animals. The conditions that were keeping the animals were totally inadequate for a lifespan study. But still, those were major findings. But that's a way better. I mean, you can see the curve is much smoother. That's a much better controlled study. And if I can summarize what we've learned about caloric restriction in 50 years, is that it works on most species, but not all of them. It works on most mouse strains, but not all of them. So there are mouse strains that if you put on caloric restriction, you actually decrease their lifespan. Which makes me think that if you take a human population and put everyone on caloric restriction, some of them will benefit from it. I mean, I can certainly think of people who benefit from going on a diet. Some will probably be detrimental. So that's my intuition from the study done in mice. It does have some negative side effects. So there's evidence that caloric restriction is detrimental for fighting infection. So I would assume, I actually don't remember for sure, but I would assume that was done in pathogen-free conditions, that study over there. Because one of the things we know is that if you expose animals under caloric restriction to influenza, their mortality is actually higher. So it does appear to have, doesn't appear, we know it has negative side effects. The mechanisms are largely unknown. It does appear to involve hormonal alterations. It's part of some sort of signaling cascade. I have one more slide about that later on. It's currently being tested in rhesus monkeys. Preliminary results suggest it has some benefits. Whether it will extend lifespan or not, we don't know. So to summarize what we know about aging, so we know that it is possible, at least to some degree, to manipulate the process of aging by genes. We know even extrapolate it to humans. Extrapolating to humans, obviously we cannot disrupt genes in humans, but they may be possible to develop drugs that target particular genes to confer the same benefits of, say, caloric restriction in human beings. I'm sure you know of a number of companies and labs trying to develop what they call anti-aging pill or caloric restriction mimetics drugs that have the same effect of caloric restriction without having to go on a diet. You know, resveratrol being the most well-known case. So before I talk about curing aging, there's anything I felt I missed in terms of like overview of field of aging. Any questions?
Well, one specific detail, maybe the naked mole rat, when you talk about...
I have a slide about it later on.
Okay.
But I mean, the naked mole rat, you see, if I can be honest, the naked mole rat was not my first choice of species to focus on. If I could, I would focus on way longer lived, one of those non-aging species like turtles. But that's quite complicated. I mean, there are a lot of... The reason we're not studying turtles is because it's difficult from a practical perspective. I mean, even cell lines from turtles, there's not that established protocols to culture them. And what would you compare them to, and how would you study them? So, in that sense, studying a long-lived mammal like the naked mole makes more sense and it's much more practical. Well, I have one slide on naked mole later on. Any more slides? So feel free to interrupt and ask questions. Go ahead.
What sort of age do sort of turtles and tortoises get to that we're aware of?
You mean how long do they live?
Yeah, in ideal conditions.
The short answer is nobody knows because they live longer than us. I mean, there's a lot of anecdotal evidence that some turtles live over 150, maybe 200 years. There's this story that there's turtles that Darwin brought into his zoos in Australia and something like that. I don't know if it's true or not. The best study I know of in turtles was a study done by Justin Compton to Iowa, I think, some park or reserve in Iowa. And sadly, it's a study that's been finished because of lack of funding, and Justin retired as well. So nobody was following the turtles. These were turtles that were followed for over 50 years. Some of them were adults. They reached maturity when they were about 20, 25. So the oldest turtle in the groups were over 70 years of age, but maybe much older. And what they looked then was age-related changes in mortality and physiology. And they didn't find any of them. They actually found that older turtles would lay larger clutches of eggs. They found one change, actually. They found that even though older turtles laid larger clutches of eggs, the chances of dead embryos or some defect in embryos were actually higher in the older turtles. So they did find that one change. But in terms of mortality and other physiologics, they didn't find anything. So these were 70-year-old turtles, which is quite remarkable, I would think, for 70-year-old turtles to still have the same physiology, or as far as we can tell from physiology, as younger ones. But if you go, I mean, one of the things we have is we have a database of longevity records in animals. I think if you go there and look for turtles, probably the record is something like 150, with a little caveat saying this is an anecdotal evidence.
So what did you say was the difference in the embryos? They lay lots of eggs, but...
They lay more eggs, but I think there's more dead embryos or dead, well, eggs that don't hatch, I think. I think there's more dead. And the first author is just incompetent. This was probably published maybe 2000, 2001. And this is one of the journals of aging, I forgot. But I can dig it up. And he retired, so I don't think he stopped. It's actually quite unfortunate because these old turtles, most of them are still alive. These 50-year-old, well, 70-year-olds, I guess. But nobody's following them anymore.
If you look outside the mammalian system, if you're looking outside the mammalian system, then why not things like jellyfish?
Jellyfish. I think that could be interesting. I mean, there's a lot of species, but as I said, I'm not looking outside mammalian field except because it's difficult to then extrapolate and find into mammals. I think Hydra is a very interesting organism, and there are studies in Hydra. I'm trying to think what's the longest-lived animal.
I thought jellyfish were quite immortal, and they are quite long-lived to almost their life cycle can loop back on itself.
I've seen those studies, and I saw that when it came out in the news, and I mean,
this is not all jellyfish, but
I know. I saw that study, and I was a bit. I would like to get more information about it. I wasn't completely convinced. I think that there are sponges in the Arctic that live thousands of years. This was done by probably the folks in Kiel, in Germany. They also have an Antarctic station. And so they looked at some sponges that were thousands of years. But sponges, they're animals, barely, So even if you study them, what you find there, I wonder if it will have applications in humans. Because ultimately, so I've always been fascinated with species with animals, and I would like to understand why. But ultimately, I would like not just to understand this process, but I would like to apply them to humans. So I'm not just curious about them. I want to use them as a tool to improve human health and extend human life. So for that, I still like to study turtles. It's just difficult. Go ahead.
I'm not really sure because I read this summer this book by Dawkins, The Theory of Genes, and in it, at some point, he made something like a theory that that is not something necessary, but it's just that genes that were responsible for aging turned out to be more successful in evolution than the others. Probably, I made an association with this fact about the older turtles bringing less children or something. So probably this is the case that.
So I didn't cover the evolutionary aspects of aging. I think from an evolutionary perspective, I think we do understand aging relatively well, because as you say, genes that, the classic theory, which I guess still holds true from Meadowmore and George Williams, is that genes that don't purposely cause aging, they weren't selected to make us age because they obviously it's detrimental, we kill it eventually. However, they escape natural selection. And also, it could happen for genes to be beneficial early in life and detrimental later in life. which is what George Williams called antagonistic pleiotropy. And so aging, how can I say, was allowed to evolve, was permitted to evolve, not because it was selected for, but because there was no selection against it. Because the major contribution to the next generation comes from young, not from old animals. And so, if you have a gene, for instance, that is detrimental at age 5 in mice never live to age 5 in the well, that gene there will be no selection against it. So it will be allowed to stay in the population. So that's the evolutionary aspect of it. And I mean, some people, James Volpel and colleagues of Max Planck in Rostock, for example, they've argued that species that can increase their reproductive output with age may be allowed. For those species, there may be selection against aging, and does explain why they exhibit negligible senescence. So that is the evolutionary aspect. I think it's well understood. So, but just feel free to interrupt and ask questions. So what about, I mean, as I said, we can manipulate it to some degree in mice, even. But that's 50% increase in lifespan. I think that would already be remarkable for humans, but that's still quite far away from curing aging. So what are the prospects of curing aging? And can we cure it in our lifespan? And I looked into my talk in 2001 in Berlin at Transhumanists, 2001. And I said, I have a slide there where I said that I was optimistic that we will be able to cure aging by the mid-21st century. I have to say I'm not so optimistic anymore. And I'll tell you why. So in April, so a few months ago, just a month ago, I gave this interview to Transhumanists magazine, Age Plus. I gave an interview to Ben Gorsel, quite well-known transhumanist and also one of the world-leading experts on artificial intelligence. And he asked me about this, and well, before he asked me about this, he asked me, what are the major findings in the field of aging in recent years? And it took me a while to answer, recent major findings in the field of aging in recent years? I honestly couldn't think of any. And this is what I said was the biggest breakthrough in the field of aging in recent years. And this is the discovery that rapamycin fed late life to mice, significantly extends lifespan. So this was done by a number of labs actually in the US. Quite comprehensive experiment and large number of animals. So what you have here, you have controls in blue and controls fed rapamycin. But the animals were fed rapamycin at this point. These are females, these are males. And as you can see, clearly there is a difference.
But 600 days, like midlife. For average.
So they were relatively less in life. And having effects not radical, about 10% increase in lifespan. So this was I thought this was quite remarkable. So it turns out that if you feed rapamycin early in life, it doesn't further extend lifespan, so the extension in lifespan is still about 10%. And so, in my opinion, this was the major finding in recent years in the field of aging. And it has a couple of caveats. I mean, one caveat, which David, we actually have a collaboration with David Harrison, who's the first author at the Jackson Lab, and he hasn't really been able to address this, which is rapamycin is an anti-cancer drug. It's also an immune suppressant, so it's not really, don't go out and start taking rapamycin, it's not going to work, it's probably going to kill you. So don't do that, because the mice are kept in pathogen-free conditions, so immune suppressant is not going to affect them. However, what rapamycin can also be doing is lowering the incidence of cancer. That's why the mice live longer. And they haven't actually been able to show that in their work yet, that the effects of rapamycin are not just because it has an effect on cancer, but rather that it has an effect on aging and multiple age-related diseases. So he has a couple of caveats. Anyone can think of any experiment in the field of aging, say, from the past 5 years that's a bigger breakthrough than this one? Because I couldn't, but maybe I missed it.
Are you talking about organism level?
I'm talking about anything. Anything in the field of aging. I think there are, and I will mention a couple of examples later on of breakthroughs related to aging, things field of stem cell and research that are more important, could have applications in aging, but directly in aging, I don't think of any.
The comment mentioned recent studies in nature, and these cells, they age, and then there are other cells around them.
Yes. So that's a study from the Mayo Clinic, from David Baker. He's the first one. And what they did was, they created, it's a very clever experiment from the methodological side, I have to say. And what they did was basically they created mice, which, when fed a particular drug, it will eliminate senescent cells from the mice. The mice are protoroid mice, that means they age faster than normal animals. They have a relatively short lifespan. But what they showed is that if you, in essence, clean up senescent cells in these mice, it will ameliorate some of the signs of aging. And it does seem to have, in some tissues, it does seem to have a significant effect. There is two caveats about that study. The first one is accelerated aging model. It's not normal mice. The second caveat, which is something they only mention in the supplementary material, just not with a business meeting, which is it doesn't actually extend the lifespan of mice. Which is not on the on the paper they say the effects of survival are given in the supplementary material, but they don't actually mention it actually has no effect on the lifespan. So, and this is something I have to agree with Aubrey. So Aubrey de Grey, one of the things he mentioned, I think is a very good point, is the major phenotype of aging is death. So if a particular intervention is not affecting survival, is not making us live longer, then why should we care about it? I do think there may be reasons to care about it. I mean, the effects, I think, one of the issues they looked was in the eye degeneration. It seems to be impressive. So if that's shown in normal mice, again, there could be applications there. But as far as anti-aging, I mean, we don't know. I mean, I guess they would have to do the experiment, which I hope they will, in normal mice.
It seems like on these charts that the drugs increase their average lifespan, but it hasn't pushed up the longest they could live by very much.
No, that's true. That's been that's true. I mean, the effect is not too much. It's about 10%. But you are right. It doesn't appear to extend maximum lifespan all that much, which makes you a little dubious whether it's again just having an impact on cancer or a specific age-related disease, or if it's actually delaying aging. It makes you a little dubious. I don't know for sure. I mean, the thing about this is, of course, that so this is the greatest breakthrough in recent years, and that's an increase in lifespan of about 10%, which is still quite shy of even caloric restriction. So, I mean, the 90s were quite exciting for the field of aging in terms of genetics. A lot of genes were emerging that could regulate aging in model systems. But I do get a feeling that it's leveling off a bit now in the past 10 years. And that's why this study, which is a very good study, but not completely revolutionary, is, in my opinion, the greatest breakthrough. Go ahead.
There was a paper just a couple of months ago. This was GSK testing its SRT-1720, which is the sirtuin drug, and it was a sample size of 104 or 108 mice per group. And that showed a longer extension, more than 10%.
That was the obese mice.
Yes, that is true. That wasn't the normal. They didn't see it normal. Yes.
So the resveratrol is, the thing with the resveratrol again is, it works quite well if you're an obese mice.
They never showed the data for the normal mice. They never fed the normal mice the drug. Yes.
There was a paper just published this week on obese individuals, I think in the Netherlands, resveratrol. And at the metabolic parameters, obviously you can look at lifespan, but the metabolic parameters seems to have some benefits. Again, whether it will work on normal individuals, to me, it's a big question mark, because it doesn't work on normal mice. Normal mice fed resveratrol don't live longer. Though we know, of course, GSK advocates of resveratrol say may have some health benefits. But again, I go back to over this basic phenotype of aging is death. Not having any effect on death. I'm not sure we should care. I mean, the other issue is that the predictive power in biology is still extremely low. I want to focus first on this example, that the rate of success of drugs in clinical trials is about 20%. Now, when a company put the drugs through a lab, I guess, put the drugs through a clinical trial, typically, well, because it costs millions of dollars, typically they have quite robust preclinical data. That means they have evidence from cell lines, human cell lines, from mouse, from rats, that the drug will do what they expect it to do. And in spite of all that, in only about 20% of the cases will the drug in humans do what it's supposed to do. And I think it's a great example of how we really don't understand biology yet very well. And we need a much deeper understanding of life to, well, not just for drug trials, I guess, but to understand aging and make predictions. And this applies to the field of very exciting emerging field of synthetic biology, which these are a couple of examples I took from a very nice review of synthetic biology. I think that's a paper published, I think it was by that. So these are cell lines, well, these are bacteria, these are cells, of synthetic biology, which is quite impressive in cell lines. But in terms of applications for humans, we really require a much deeper understanding of biology. We must be able to do what engineers do, which is understand all of the individual components, understand what happens if you put them together. And that's something we still don't do. Which leads me to my next slide, which I just felt I had to mention, about Aubrey, which I know he gave a talk here. That's actually a picture he sent me when he gave a talk which I hosted in Liverpool, which I think is the only picture, at least I've seen, of him in a suit. So.
He wore the same thing in a recent talk that he gave in San Francisco.
Okay.
Like 3 days ago.
The same one?
Yeah, pretty much. It was the same getup.
Well, I thought he was a nice one.
I think he wears that whenever they pay him to talk. Something like that.
We didn't pay him to talk. So presumably, maybe not all of you, but most of you would be aware of these strategies for engineered negligible senescence, in which, for example, it says that there's a 50% chance that we will be able to control aging in the next 25 years. And I thought I would, well, actually, first of all, how many of you agree with that statement?
What do you mean by controlling aging exactly?
What do you mean? I mean, it's decided in the same sort of medical control that we have overcome most infectious diseases today.
Isn't it like principle?
Sorry?
I'm sorry.
Where have you got that decision of responsible? It's like, you know, if they do one, it's already successful. Yeah, you can control.
Well, no, but I mean, it's bringing medical control.
Yeah.
So you would have it at the clinical level, not at the basic science level. So you'd have that available to everybody.
Yeah. So you'll be able to, just like you take antibiotics for bacterial infections, I guess. You take some drugs.
Alcohol.
Okay, so maybe.
Laying it somewhat, yeah.
Sorry.
Certainly the early treatment.
You think maybe it's 45%?
No, I mean, I think that we might have access to sort of early treatments that allow us to delay it a bit, and then obviously it'll allow us to get more during the meantime.
I see. You'll get like to what he called escape velocity.
Yeah, possibly. But I don't think we'll be able to like cure aging in the next 25 years.
Again, it depends what you mean by aging, because if you're very interested in sort of stopping aging at an early enough stage that I mean, people are living longer and longer anyway, just because they're healthier and so on, better medical treatment. But then you're living longer in your old age, like you get to the 60-year-old physical state, and say a 60-year-old physical state, for about 30-year-olds.
That's the point I wanted to make with the Rapid data, is that you can't consider death as the only phenotype, because rapamycin was also compressing morbidity, or which is reducing the amount of time that you spend as a decrepit man or a decrepit human. So that also is beneficial to society. So
that's true.
Yeah, that's because it'd be better to live to 80, everyone lives to 80, no older, but you stay completely healthy and live to 30 or 40 until practically until you die. And that'd be better than being, you know, in a bed
In a
10 years,
decrepit
then dying
state.
when you're 80.
Well, yeah, that's true. But I think what Aubrey means is reversing aging. So what we can do in infectious diseases is if you have bacterial infection, you can take some antibiotics, and it will reverse. You'll be healthy again. That's what I think he means, because he talks about reversing aging, the rejuvenation technologies. That's what he means. And for that, you actually need to complete what he calls a decisive level of medical control of aging. Now, I think most researchers have considered that components of the proposal optimistic. I mean, there was a debate in EMBO reports. Hubert Werner, he's the first author. There's a number of scientists, not me, I didn't sign it, but there were a number of scientists who basically wrote his report against Sands, basically saying that it was fairly optimistic. Aubrey responded to it, so it was actually an interesting debate. So if you're interested in Sands, I do recommend reading Werner's review and reading Aubrey's response.
Well, I thought it was 16 of the scientists, practicing scientists with Aubrey who responded. There was 18 scientists signed that, and I think 28 scientists signed the response.
If I remember correctly, the response was just him.
Ah.
That there is another. I mean, there's I think a commentary in another journal where he's a co-author with other scientists. But the response to Werner, I think it's just him. And they had another debate actually in Rejuvenation Research, which is the journal Aubrey is editor of, between Aubrey and Hubert Werner. And I mean, basically, I think my argument, and this is something Alex argued actually, is that, well, if we could engineer biology, if it was so simple, then surely we could cure cancer, because cancer, if you think about it from an engineering perspective, it's all about removing undesired cells. That's all we need to do. And of course, in practice, it's much, much harder to do that, because cancer cells are just hard to discriminate from the others at the molecular level. You can look at them in the microscope, but to develop a drug that will discriminate between them is actually extremely hard, which is why most chemotherapy treatments are actually drugs that will kill a lot of healthy cells. And they just work on the assumption that cancer cells are going to be less resistant. And of course, having said that, I mean, the success of SANS depends on future technology. So Aubrey's argument is that, well, maybe we don't have the technology yet to cure cancer and to engineer biology, but it's imminent. We'll do it in the next 25 years. And by then, we'll not just cure aging, but we'll cure cancer and a number of other diseases. Which I personally think is optimistic in the sense that, well, there are future technologies, we don't know if they're going to pan out or not. Having said that, I do think Aubrey has done a tremendous work in attracting attention to the field. And he did one thing which I thought was remarkable, is that he attacked the idea that aging is untreatable. It's a big shift in the field, because, I mean, I've actually known Aubrey way before SANS, since I was an undergraduate, and he back in the day, I'm sounding like an old man, back in the day, when I was a young man, scientists, even if they believed in it, they wouldn't talk about curing aging or treating aging. And nowadays, there's still a number of scientists who oppose this idea, but there's at least some scientists who talk about it. And curing aging, I think there was a review by Campisi and Big on curing aging in publishing Science. It ended up being rather conservative than if you read the text, but at least it's up to discussion, which I think is something remarkable. I mean, it attracts attention to the field, and in that sense, I do think it's been very positive as an initiative. And then, you know, maybe, obviously I don't know the future, but maybe some interesting outputs will come out of some of its components. So, now having said that, I do think aging is curable. Not sure it's going to be in the next 25 years, but I do think aging is curable. And so what I'll tell you next is some ideas and a couple of projects from our lab on how I think we can go about this. And the first point is, if you think about it, then again, you see 2001 was a very exciting year to do science. The human genome was sequenced, and if you think about the human genome, this is the digital code from which the machines of life, the proteins, emerge. So if you can decode, if you can decipher the genome, we can then understand aging, we can then try to manipulate it. Then we know, of course, that a tiny part of the genome can have a great impact on aging, because single gene manipulations can impact on aging. Now the problem, of course, is that our understanding of the genome is still very incomplete. So without going into great detail, the genome is big, 3 billion base pairs, and I actually don't know how many now. I'd say it's less than half, or almost half of the roughly 20,000 human genes are still poorly understood. There's a number of genes we don't know their function yet. And in addition to genes, to protein-coding genes, there's a large array of regulatory mechanisms, microRNAs, other RNA genes, epigenetics, etc. So there's still a lot about the genome that we don't understand, and there's done a lot of Ramsell called the unknowns, the thing we don't know we don't know. That may still happen. So what I call we do is interpretive genomics of aging. And I'll tell about a couple of our projects, because the idea was that our lab would do research, basic research on aging, but also from a genomics perspective, because I think we need to understand, I think we need to tackle the complexity of life, including all the genes, understanding all the genome, and modeling cells as a whole before we can actually tackle aging. And so what we've been doing is really trying to work as much as possible on a whole genome basis. So sometimes you have to focus on specific genes, of course. So for example, this was something I did some years ago. We created a database of genes associated with aging and longevity. There's over 1,000 genes now associated with aging in model systems. These are individual genes that if you disrupt, knock out, overexpress, whatever, it has a significant effect on aging or lifespan. And the point of this database, which I call Genius, is that it's a resource for functional genomics. That is, people doing genome-wide approaches, genome-wide association studies, genome-wide gene expression studies, they could, if they were interested in aging, or even if they just had a slight interest, they could take GeneAge and then use this set of genes known to affect aging, and they use it in their analysis. And it's been, I'm not going to talk about, but there's been a number of studies where researchers do exactly that. They just take our GeneAge data set and then link whatever study they're doing, whatever analysis they've done, whatever data they generated, they try to find links to aging using our data set. Another thing we do at a bioinformatics level is just take advantage of the massive amounts of information. So this is from Geo. This is the number of, so Geo is primarily a microarray repository. This is, as you can see, the number has been increasing. There's hundreds of thousands of records now. So one project, and this was done by a PhD student in our lab called Pedro de Magalhães. What we did was create a co-expression map. So the idea is to, for all the genes in the genome, quantify how strongly they're co-expressed with each other. The idea being that genes co-express, that in genes that tend to go up at the same time, or go down at the same time, they tend to be functionally related. And this can help us derive candidate genes, derive putative gene function, identify disease genes. I'll give you one example. So for we employed over 1,000 data sets, over 20,000 samples. And then what we have is a matrix of 20,000 genes by 20,000 genes of how strongly they co-express with each other. So what it means is, so these are gene symbols. What this means is that when this gene, say, goes up or down, in 71% of the cases, go to this one. So that gives you a strength of how the genes are co-expressed. And one of the applications is actually looking at unstudied genes. Because as I said, for almost half the genes in the genome, we don't really know their functions, and I think that's a major issue. So we can derive putative functions for poorly studied genes by looking at which genes are co-expressed with them. So very simple. If you have a gene you don't know the function of, you just look at which genes are co-expressed with it, and you then look at which functions are associated with those genes. So, for example, for this particular annotated gene, this is just a single point phrase, we can see that the genes that tend to be co-expressed with it tend to be associated with the mitochondria. So we can say, well, this is putative gene related to the mitochondria. And we can then test experimentally. We're actually not testing this one, but we're testing another one which we think could be related to cancer. And we're doing some experiments in cells, and it seems to correlate with our predictions. So another thing we've been doing again on a genome-wide basis is deriving a signature of caloric restriction. And the idea is that by aggregating essentially all microarray data we could find on caloric restriction and analyzing it as a whole, we can try to identify what are the crucial signaling pathways in caloric restriction and identify hubs and possible new targets for drug discovery. I'll say one thing, however. So some transhumanists have actually asked me, why are you working on caloric restriction? It only extends lifespan by 50%. I mean, we're not going to cure aging just by looking at caloric restriction. So why do you focus on it? And I think that's a good point. But the reason is that, well, true, I would like to do something that would allow me to cure aging. But we also have to do analyses that are practical and that could lead to results now, and that are fundable as well. And caloric restriction is a well-established paradigm. And the truth is, if we can identify new targets for drug discovery, if we can identify new drugs that can manipulate aging via caloric restriction mechanisms, then we already have a tremendous benefit on human health, even if it wouldn't cure aging. So I won't go into great details, but basically what we try to do is we try to identify, this is being done by Michael Planck, who's sitting over there, so if you want more details, you can ask him. That's him over there. I'm not going to ask you to stand up and give a rousing ovation, but so without going into great details, the idea is that so we got to conserve molecular signature of caloric restriction, and then we try to look for, we can look at processes, we can look at functions related to caloric restriction, but what I'm really interested in is what are the key regulatory hubs. And you can look at transcription factors, you can look at networks, do network analysis, try to identify which proteins are more important in caloric restrictions. So for instance, we have this model, I won't explain it all, but so in blue you have proteins genes we found underexpressed, and in red proteins we found overexpressed in caloric restriction consistently across tissues and species. So there are some transcription factors like SIRT1 and RA, which we think are important in the signaling cascade of caloric restriction. So that's the aim. The aim, I guess, is trying to decrease the complexity, because obviously when you do a genome-wide study, you have lots of genes changing. Try to decrease the complexity of the process and try to focus on some key regulators. We're also interested in trying to integrate data from various models. We know there's gene expression changes in aging, there's physiological cellular changes. So for instance, we developed this portal called the Digital Aging Atlas. The idea is to have age-related changes at different biological levels, sometimes with causal links, to try to allow researchers and clinicians to get a more holistic, more integrated view of aging. You can look at different organs, etc. And finally, as I said, I've always been fascinated with species that appear not to age. And I'm also interested in the variation of longevity from mice who live up to 5 years, all the way to humans live over 100, and the longest lived mammals, which are the bowhead whale, which have been estimated to live over 200 years. So I've always been fascinated by this as well, and trying to understand genes, trying to identify genes that are important in this. I should say the differences in longevity between mammals are not due to metabolic rates. That's the rate of living theory, which has been around for quite the first half of the last century, I guess, which makes some intuitive sense. Species with higher metabolic rates accumulate damage faster and are going to age faster. I'm not going to show the results, but we and others have disproved that. And so what we're interested in trying to identify genes that determine rate of aging. And again, the goal is that if we can do this, then this may have human application. It explains why humans are susceptible or resistant to particular diseases. Okay, it can help us make us more resistant to certain diseases. We get a number of researchers that look at parameters that correlate to longevity. We've done some analysis, for example, of mitochondrial DNA. We're also looking at comparative genomics and trying to identify genes that may be associated with long-lived species. And Young Leaf, who's sitting over there, was formerly in our lab, has also done some work on that. It's him, the one sitting right next to Michael Planck. And since you asked for the naked mora, so if I could, I would like to study bowhead whales. That's what I would really like.
How do you get them in our lab?
That's what my boss told me. So we couldn't really get them in a lab. So I guess our second choice was the naked mora, which you may have heard is the longest lived rodent. But to me, the most fascinating aspect of the naked mora is that it's cancer resistant. And I mean, the genome was just published by Vadim Gladyshev and Colin. We're just doing some transcriptome sequencing as well. For example, we found that mitochondrial oxygen reduction genes seem to be overexpressed in the naked mora. When compared to mice, and how exactly that would impact on aging, we're still not sure. We're doing cell studies as well. The thing I would say about the naked mora is that, of course, it has a great advantage that you can compare it to short-lived mice. So that's why I think it makes it a good long-lived model of aging. Because obviously bowheads would be wonderful, but then what would you compare it to? How would you study that? You could try to get cell lines, I guess. Although there are legal issues with that, which I won't go into. But the naked mora, I think, is a great model because you have a long-lived rodent, which you can compare to mice. So you can do that comparison. You can compare it to mice and rats. And then what you find in naked moras, you can try to test in mice. So for example, we're cloning, this is one of the projects we have with David Harrison at the Jackson, which is taking naked mora genes and putting them into mice and seeing what happens if the animals live longer. So in that sense, I think it's a really cool model. Literally speaking, because they have a lower body temperature. So, any questions on? Oh, actually, I have one more thing to mention, which is this graph. So, I'm sure you're aware DNA sequencing costs have been decreasing dramatically. I mean, even faster than computer power. That's George Church, who was my postdoc mentor. And I mentioned Alcyon Molecular because they're quite interested in aging. Their idea is to make biology digital. And I do think this is a great opportunity, not just for biomedical sciences, but aging in particular, because you can, as Alcyon aim to do, we can turn biology digital. We can just do genome-wide studies, patients in a number of conditions, gene expression, and get a large amount of data, which you then need to analyze bioinformatically. So one of the things, for example, we're doing is sequencing the transcriptome. And one of the things it's not published yet, but we've seen is in aging rats, if you use RNA-seq, if you sequence the transcriptome instead of doing a microarray, so RNA-seq is much more sensitive. And not only that, but it can detect splice variants, it can detect RNA genes, it can give you way more information than a simple microarray. And so, for example, there's not that many genes differentially expressed with age. We observed that with RNA-seq as well, so just like in microarrays. But then with RNA-seq, we found a number of splice variants that change with age. We detect RNA genes. We even detect novel genes that weren't annotated yet. And so the idea is that biology can become an information science. which again would then lead to much higher prediction power. So my kind of roadmap to aging is that we need a deeper understanding of biology and the genome in particular, the genes and their interactions to understand, to be able to manipulate aging. And we need a better understanding of the methods of aging. So this is what our lab focuses on. There's another approach, which is not what we do, but I'll just mention it, which I still think in terms of curing aging, I do think we need better methods to modify biological processes in people. I mean, there's obviously drugs, we're working on it, but that's still quite limited. I mean, the point is, there are a number of diseases for which we know the pathophysiological process that we still can't cure. Neurodegenerative diseases, I did my internship when I was an honors student on a neurodegenerative disease called Chagas, fairly typical in Portugal, which is just the accumulation of this defective protein, which then kills neurons and eventually kills the individual. So we know very well what happens at the molecular level. We still cannot intervene with it. So we do need new approaches to modify processes in vivo. And I do think in terms of aging, I'm not going to run you through all of this. This was a video I published a few years ago on the Immortality Institute book. The idea is that to intervene on aging, Sam Gossip from Neverness, which is a book from David Zindel, which is an entity that essentially allows us to rejuvenate. But the idea is that even if we know what causes aging at the molecular cellular level, to actually intervene in it, we still cannot do it. I mean, there are technologies that are in the development, like stem cells, like gene therapy, but they still are not there. So this is the other component I think we need in order to cure aging. I mean, certainly in terms of stem cells, there's been a number of successes, like induced pluripotent stem cells. So I think in my interview with Ben Gorso, I said, well, there's not been that many great findings in the field of aging in recent years, but certainly induced pluripotent stem cells. I think it's been a major breakthrough. It was initially published in 2006 by a Japanese group, and I think they'll win the Nobel Prize for it. Foster a new field. So I think for those who don't know what induced pluripotent disease is, the idea that you can take adult cells and you can reprogram to become pluripotent cells, which can then generate differentiate into essentially all of different tissues, which can be used for regenerative medicine. For example, this is a strong component of the SNAP approach. I mean, there was just a paper published by the French lab this week on induced pluripotency of cells from very old individuals. And they show that it reprograms them and, I was going to say rejuvenates them, but yeah, I guess you can say that from the acid that they use, it rejuvenates them. So this is certainly an incredible technology. Any questions before I move into my backup plan? on aging, curing aging.
What sort of factor or IPS3 programming factor?
I think initially it was 4 transcription factors, OX4, SOX. It was a violent cocktail of 4 transcription factors that we used. But now there's other methods as well. I think I've seen a method with. So it's because the issue is that well with viral factors, then you have cells infected with viruses, which creates issues. So I think that there's now other methods. I mean, there's still a lot of questions. I think there was a recent paper showing that, well, induced pluripotent cells, they're not perfect. They can have problems in mitochondria, for instance. So there are still issues. There's still a lot of work left to do, but there's tons of labs and companies working on it. So I do think it's a very exciting area.
Yeah, you can change them now, adult cells, by just in a differentiating media. So you can have cytokines in the media and you can change them directly. You don't have to, and also microRNA that you were pointing out. So yeah, so
there's a number of methods.
Yeah,
it is quite remarkable. I think again, there's a lot of work still to do to turn this into clinical practice, but it does hold a number of.
Something that does seem to have come out in recent years is the extent to which evolutionary processes keep carrying on even within our own bodies. There is, of course, the model of cancer as a kind of evolutionary process. that can't be taught. There's a very recent discovery people may have read about, apparently our brain cells are genetically distinct from one another.
No, I don't think I've heard about that.
I never did. It recently hit the semi-popular Life Science Online.
I'll look at, so brain cells are different than what they compete with each other?
Well, no, not in the sense of competing with each other, but when you look at their DNA and the expression of it, they seem to vary far more than cells should.
I see.
So not that simply identifies a useful evolutionary process going on. Nobody has a hypothesis, I think, as to why it would be useful for brain cells to actually differ from one another. But anyway, certainly in cancers, certainly in what happens with mitochondria as time passes, you do observe that it seems very difficult to stop some kind of evolution. When we look at colonies of social insects, there is the problem of mutiny, and suppressing mutiny does seem to be what stops things like termite mounds from getting kind of even bigger and more cooperative, as it were. Now, if there's a kind of fundamental evolutionary limitation on how long an organism can live without internal competition somehow getting out of control, that would be a very fundamental problem.
So first of all, I think that's an interesting point in terms of evolution, internal evolution. It's actually relevant in terms of interventions. You're thinking that if you can build stem cells that have an evolutionary advantage over existing ones, the old ones, you can then make evolution work in your favor for rejuvenation, for instance. Which I think is something I mentioned actually in this review. Of course. Now, in terms of that, well, I think one issue would be, say, and this is to me a big mystery, is how an organism like the bowhead whale, which can live over 200 years and is massive, doesn't get cancer.
Yes, you're familiar with Peto's paradox and work done in...
So that to me, that's a big question. So it's a good point, but I don't know why. No,
So, Roy knows all about this. The multiple copies of the P53 gene discovered already in some big animals. Of course, sorry, I know less about this than other people present, but there is the question, of course, as with the mole rat, if you can evolve an anti-cancer mechanism, why doesn't everything have it? In the naked mole rat, we know that there is a hypersensitive contact inhibition, seems to be a key mechanism that stops solid tumors forming. But given that...
We don't know for sure, it's an hypothesis, but yeah.
Okay, well,
we have
respectable the investigator's hypothesis. The question of why don't other animals evolve that? Peto's paradox, why very large animals with huge numbers of cells don't get far more cancers. A tentative answer is apparently multiple copies of the P53 gene in each cell. But if it's as easy as that, why doesn't everything do it?
I guess we go back to the evolutionary issues that we don't have the anti-cancer mechanisms of the bowhead whale because we spent our lives, if you look at human history, we didn't live this long. I mean, our average lifespan 1,000 or 2,000 years ago was 20 years. Few individuals reach old age. So there was evolutionary no reason for us to evolve anti-cancer mechanisms at the level of the bowhead whale. So we just don't...
I mean, I'm not entirely convinced. I've heard this argument very often from medics and biologists. But for example, rodents suffer cancer a lot. Yes. Even humans in ancient times didn't, you didn't never get cancer in your reproductive years. There was always, it would be useful to get no cancer or cancer later. We know, for example, from menopause, from grandmother hypothesis, even in later life it would be useful if humans stayed healthier longer. So, and nobody can find, in the case both of Peto's paradox, of animals with huge numbers of cells, and in the case of naked mole rats, there doesn't seem to be a cost that anybody can actually find associated with anti-cancer mechanisms. The hypothesis has always been a good anti-cancer would just be too metabolically costly, or something like that, for it to evolve. And yet those creatures that have evolved it don't seem to be paying any cost. Not easily findable.
I think that's a good point. I mean, I don't know how much pre-maturity or pre-reproductive mortality of cancer was in ancient times.
I don't know about humans, but certainly in rodents, they get cancer so much.
Even wild-derived strains. Because remember, the laboratory strains, they're
highly inbred.
highly inbred, so they probably
I
get
think
more
wild rodents
cancer.
get cancer a lot too.
The wild-derived, they get cancer. I can tell you from Steve Ossed's lab. So Steve Ossed went around the campus in Moscow, Idaho, looking around old barns and things to capture all the wild-derived mice, and set up this population of wild-derived mice, which live slightly longer than laboratory strains, and they do die of cancer. But I don't know about the young animals. I mean, I presume some of them, but things can always go wrong. Right?
Yeah, and rodents die so early, correct me if I'm wrong, but they are actively breeding typically until the very end of their life, aren't they? I mean, they certainly don't get open like the menopause, or there's no cut-off then.
Yeah. So, I don't know. I don't know evolutionary. I mean, it can be quantified. Maybe some other people have looked into it. What are the costs of evolution? I mean, from a fitness level, from a selection level, based on how many individuals died pre-reproduction, how he would affect it from an evolutionary perspective. My guess would still be that it's relatively few. And again, it would be the evolutionary pressure would not be sufficient for the animals to evolve and indicate.
Indeed, but I've heard this expressed as a guess very often, and yet actual hard data, very, non-existent on what does it cost the mole rat to be cancer resistant, what does it cost large animals to resist Peto's paradox. I don't think anybody has any clue really.
I don't know that. So I'll just tell you the last thing about giving a talk to transhumanists is, of course, that I can mention things that I normally would in a normal scientific conference. So the last two things I want to mention, first of all, is artificial intelligence. And the reason I mention this is that it has been pointed out, I've actually had quite a few number of students visit my website and contact me with an interest in aging, who then argue, well, maybe we're just not clever enough to tackle aging. So what we really need to do is create an artificial intelligence, and then the artificial intelligence will cure aging. And again, I've met more than one person making this argument and working now in artificial intelligence. So I don't know if anyone here works on artificial intelligence. But it does showcase a variety of ways people can contribute to cure aging. If you can find a robot that can do my job better than I do, I'll happily resign. And the last point I wanted to mention is cryonics. And I saw on our website you have a talk from whose name?
Cryonics.
What's his name?
Yeah, I know exactly what you mean, but...
So the guy from Manchester who works in cryonics. So, I mean, the concept of cryonics, cryopreservation, is very simple, is that, well, if you die, and unless you believe in God, death is eternal oblivion if you are buried or if you're cremated. Well, on the other hand, if you're frozen, there is a possibility of future revival. And I thought, well, the issue to me is that the current protocols of cryonics do have serious limitations. I think that a frozen cell, and when you freeze cells, you can see massive damage and mortality. And if you freeze the whole body, you're going to see a 2 much wider degree. Though actually you don't freeze bodies, you vitrify them, even so. But however, in partly inspired by one of my former students, Lauren, who's now in London, we started looking into it. One of the things I've actually noticed is that cryonics, biostasis, reversible biostasis, that is freezing someone and then reviving them, could be achievable with current technologies. In other words, yes, there are serious limitations to the protocols, but there may be just the right chemical formula, the right combination of chemicals that allows us to avoid vitrification damage and allows us to vitrify a whole human being. I do think that, well, for instance, aging, I do think it's something that in order to cure aging, we will need to develop new technologies, stem cell therapies, etc. I do think cryonics in that sense, I think it could be solvable with current technologies. And the other point is that there is a huge commercial potential. I mean, it's not a very popular topic, but if you could actually develop reversible biostasis, freeze someone at a particular point in time for a limited time, and then revive them successfully, it would have tremendous commercial applications. I think it would be one of the big industries of the world, because think of all the people who die in hospitals. If they were given the chance that when they have a little pathology that they are frozen, I would think if they have the means, I would think they would go for it. And so this is actually something we're exploring with memory companies. It's a topic that's not very easy to get government funding for. But we're exploring this with companies on trying to improve on the current protocols of cryopreservation, ultimately to develop a viable reversible biostasis. So actually, is anyone here signed up for cryonics? Okay. But does anyone believe that it could work? I mean, if you were told that you would die tomorrow, would you sign up?
I believe it could work. There are huge risks that have been explored in science fiction. What kind of world will you be poured out into?
Right.
It's better than being eaten by bacteria.
Right. It's better than eternal oblivion, basically.
That's
doing something called a mock that lives in the Arctic, it takes 10 years to transition to a button party to live each season with enough good food, and then it freezes just as the cancer cells for 10 or more years before the party when it's transformed. Yeah, it's still not strong. There must be some biological mechanism.
Yes, so I did see this. I saw that show actually, and there are some species that do appear to be able to freeze way better than us. But they can secrete cryoprotectants. So there are certainly insects that can do it, and they can secrete cryoprotectants. Their cells are very prepared. I think it's quite interesting. I think how do you apply, even if you find out that how they do it. For example, I think there was a paper on the membranes of the cells of some insect that were just different than ours. So that's, I think, interesting, but that's, excuse me, not easy to apply to humans. Having said that, I think it is, again, quite remarkable that some species appear to do it. Again, going back to the species of senescence, animals that fly, and the Wright brothers. So in that sense, I think it's an inspiration. Animals can do it. If we figure out how they do it, maybe we will be able to do it. So I mean, like I guess most of you, I do have some serious concerns about the current protocols. But I do think it could be achievable, and certainly, and it is an area we're interested in. I think that's it. Right, so I told you that aging is remains a mystery of biology that most likely will kill you. We can manipulate aging by drugs and genes to some degree, but it's still, when you think about curing aging, it's still quite far off. But there is reason to be optimistic. I mean, genomics is turning biology into a digital problem in which we can tackle the intrinsic complexity of biological systems. And I'm very optimistic about it. With the lowering cost of sequencing, with Moore's law and increasing computing power, that we can start to tackle the complexity of biology. And from there, I do think that we will also, in addition to understanding biology, understanding aging, we will be to develop new interventions in vivo in order to cure aging. And so I didn't include an acknowledgement page because I mentioned so obviously a lot of what we do is like people I mentioned Young and Michael and Cisco and Sean, a number of people from our lab, and we are funding from a number of research councils and charities. But because this was a transhumanist meeting, I thought I would mention Robert Bradbury. Did anyone here meet Robert Bradbury? So he was a well-known transhumanist, life extensionist, who passed away earlier this year. And he was the first person I met, back in the day. Again, old man talking. Back in the day when the internet appeared, he was the first person I met who also wanted to cure aging. So I thought I was the only person in the world who who wants to do this. And nobody in my family was think that sounded that excited. He was the first person I met who also wanted to cure aging, and he actually had a company at the time called Helios Corporation who were working on this. And so he was a great inspiration, and sadly passed away earlier this year. But I do try to follow his motto that was something he told me, I think, maybe 15 years ago. So I hope you learned something and to try to have some fun. So, thank you very much.
Okay, we'll have a few questions now, I think, and then some of us will offer the pub for a quick half before we go train back. So if everyone has any questions. Well, I definitely have a question. So on Tuesday, the Mass Society also had a talk on aging, and one of the things which came up was the idea that you have genes which are advantageous when you're young, but the cost of being disadvantageous when you're a lot older. Such an environment isn't really a problem because you won't reproduce that point anyway. So the idea was that maybe you'd have lots of genes whose problems all line up and occur at the same time. So the problems that occur after you would have died aren't a problem from the evolution point of view. So you might have lots of genes that all cause problems that all hit at the same age. So curing aging would be very hard because you solve one of these problems that took a bunch of others at the same point. You wouldn't really make any progress.
I think that's an issue. I mean, the truth is, I do think in terms of aging that there is some synchronizing mechanism, some common process of aging that coordinates age-related changes to a large degree. Having said that, I do think that some age-related diseases could have independent mechanisms. And the best example I can think of are neurodegenerative diseases. I mentioned Machado Joseph, but that's very common, but Alzheimer's disease and amyloid deposits, which could be independent. In other words, they're the result of this late-acting deleterious allele. They're not really related to aging. They're just due to some process that goes wrong at very late ages. So I definitely agree that could be an issue, which again is why we need to. But if there are those, then we have the understanding of genomics, and we have the understanding of biology, and you have the means to intervene in vivo, then we may be able to tackle that. I actually, the first pseudoscientific paper I've ever published was a paper whose title was Postmortem Lethal Genes. And the idea was that there may be late-acting alleles after our maximum lifespan that start, so there may be unknowns. There may be processes that are going to kill us, even if we extend lifespan. So I definitely think that could be an issue. So that's a good point.
In your backup strategy, you haven't really mentioned a piecewise approach. For example, to cure baldness, starting to really trivial level, you can grow skin, young skin. There's a real possibility of doing a skin transplant to cure grey hair and baldness. There are individual organs that you could probably grow afresh, a young version of the organ. The extension, to the science fictional level would be the whole body transplant, whereby a bit at a time you can replace bits of yourself with much younger but identical genome. I'm curious you haven't mentioned that one, because that's maybe nearer to what is currently being approached than any other.
Near the degenerative diseases, can you ever replace the entire brain?
Especially if the brain does turn out to be the most vulnerable point. However, neural tissue can, in many people, outlast the rest of the body or bits of the rest of the body. So,
True. I think.
I suppose you're talking about regenerative medicine.
Well,
I'm really talking about growing new, young organs and transplanting them into you.
Yeah,
instead of just stem cell therapies, or. But that's more. Transplanting organs is more invasive. It has that advantage. There are efforts along those directions. I think the Methuselah Foundation has what do they call it? New organ prize or something along those lines.
Ancestry stuff to do it anyway, like people who have liver and kidney transplants, you know, domain death.
Yes, indeed, but normally from a donor who has a different genome to yours,
Yeah.
whereas this would be,
Your own.
from your own
Yeah.
stem cells, whatever. Person replacement.
I think they could be useful. My intuition is that that is quite invasive. So if there is a less invasive way of doing the interventions, you know, a stem cell therapy, you know, with genetically engineered stem cells that will replenish and rejuvenate a particular organ, I would think that would be preferable to just replacing the whole organ, because it's less invasive, and there's always issues with cutting someone open and replacing an organ. So that would be my intuition. Having said that, I think there could be a possibility. You say growing organs again, but there's also an emerging field in artificial organs. So there are possibilities there in terms of rejuvenation, which I certainly think could play a role.
Just one thing to add to that. Just earlier this week, the engineering department had a talk by, do you remember the name of the person? She was from Imperial, and so they're working on regenerative medicine, which is tissue that is made from that IPS process. So take your skin cells, take a scaffold, just basically the scaffold of the organ from pig or something, discarded organ, and then you can just spray it on with your cells, and you can, it's sort of like a bladder or a kidney or so forth. So Imperial, Wake Forest University, which is where SENS Foundation has its collaboration with Anthony Atala in Wake Forest University, who have already transplanted these kind of organs, and McGowan Institute, which is also in the States. So those three places are already making organs that are for humans. The bladder and kidney are the two so far.
It could be an area of potential, no doubt.
The low-hanging fruit.
Yes. Anyone else? Did you have a question?
It was actually pretty much on that anyway. I was going to say about the brain. I think there's lots of questions on that, but one thing we can't prevent is the brain. So we have to be truly preventative.
Yep. But we don't know, I think, for example, we don't know the degree to which brain aging is intrinsic or is derived from extrinsic factors. So, for example, the immune system ages. We know that, and we know inflammatory levels go up with age. So that, I would guess, also plays a role in brain aging. So if you were to keep your whole body young, I would think, my assumption would be that the aging of the brain would also be delayed. I don't think it would be stopped, so we still need to do something to it, but I think it could be delayed.
Okay, so I think we'll round up there. So to come back next week, we've got lucid dreaming, which should be very good. But in the meantime, let's join you once more. Thank you.
Thank you.